We're in the cell organelle. And in this lecture, the main focus is know each organelle's name and its function. Okay, let's get started. So inside the cell, you have a lot of organelles. It's like in your body, you have a lot of organs. Each organ has their function. So each organelles have their function. The first one we're going to look at is ER, endoplasmic reticulum. So ER, its structure look like a blanket. It surrounds the nucleus. So that's the nucleus outside. These are the ER. And there are two kinds of ER. One is called the smooth ER. That's the smooth ER. And this is called the rough ER. And the rough ER is rough because they have those ribosomes. So those blue dots are ribosomes. And let's look at the ribosomes function. Ribosomes, part of its function is for the translation. Translation is from mRNA to protein, so that's ribosome. And ribosome provide a space for the translation to happen. So part of the ribosome is rRNA, ribosomal RNA. So it provides a space for the translation. Translation is from mRNA. So this string is mRNA to protein, to amino acid sequence. And you have the tRNA. This is the tRNA. There's three kinds of RNA. Ribosomal RNA is part of ribosome, mRNA used for the genetic information, and the tRNA. T means transporter. Transporter RNA gonna carry the amino acid. These both are amino acid to here and for the translation process to happen. So it happens in the ribosome, and ribosome located in the rough ER. So that's the rough ER. Its function is process, sort protein. So translation process. This is translation process. After you have the mRNA, and then you have ribosome to help translation, and eventually you create the amino acid sequence. And once you have the amino acid sequence, technically it's not a protein yet. So protein need to have its own 3D structure to perform its function. That's why the amino acid, we call this amino acid sequence, need to be transported to other organelles and keep being folded to its correct 3D structure, and in order to perform the protein's function. Okay, so again, this is the rough ER. This is the nucleus. Outside the nucleus, that's the rough ER. And you also have smooth ER. And the rough ER has ribosome. That's why they look rough. Its function, protein synthesis. It creates the amino acid sequence, or we say the translation process happens. You create amino acid sequence. And those amino acid sequence need to be transported by vesicle to another organelle and to be modified there in order to create a functional protein. And then they will be excreted out by exocytosis. Okay, now let's look at the smooth ER. Smooth ER does not have ribosome. They look smooth under the microscope, and they still connect to the rough ER. So this is the nucleus, this is the rough ER, and that's the smooth ER. Okay, let's look at smooth ER's function. Smooth ER does not have ribosome. So its function is not to produce protein. It produces anything else, lipids, carbohydrate. So if your cell membrane is damaged, that's the smooth ER's function to fix it, because cell membrane is mainly made of phospholipid and some protein and some carbohydrate. It also detoxifies poisons. And the third one is important function for skeletal muscle. Skeletal muscle is smooth ER turned into a calcium container. because in order to contract the muscle, you need calcium to trigger the muscle contraction. So in the skeletal muscle, their smooth ER turn into the calcium container. So when the muscle contraction happens, they're going to release all the calcium to trigger muscle contraction. Okay, next organelle called the Golgi apparatus. Golgi apparatus is around here. So it's outside nucleus, rough ER, smooth ER. Now they go to the Golgi apparatus. Its function, still protein synthesis. We have the mRNA come from the nucleus, go to the rough ER, and rough ER has ribosome, so it helps to produce the amino acid sequence. So we call the translation process. But proteins need to maintain its 3D structure to have its function. So after the rough ER produce the amino acid sequence, those amino acid sequence need to be transported to the Golgi apparatus and keep being modified. They will be folded to the correct 3D structure, and now it's protein. And the protein will be transported out by vesicle exocytosis. They can be released. So Golgi apparatus is still part of the protein synthesis function. Next organelle is called lysosome. Lysosome is function is like a stomach in our body. It digests. So it has digestive enzymes, those digestive enzymes. When a cell, endocytosis, swallow the bacteria or food in, and they will move the lysosome there. And because the lysosome have the digestive enzyme, they can digest the food or break down the bacteria. And during the embryo development, the embryo, when the hand develop, it will grow up like a big fan. And between finger and finger, those cells need to be digested away. So that's part of the lysosome's function. So our hand development is not one finger, each one extend out. It's actually a fan grow out. And then between fingers and fingers, those cells will be digested. And that's also lysosome's function. And also white blood cells, they digest bacteria. It's also lysosome's function. So there's a lysosome. Inside have digestive enzymes. When white blood cell swallow in phagocytosis, they swallow in bacteria, and they will use lysosome to digest those bacteria. Next one, a very important organelle called mitochondria. Mitochondria has double membrane structure. So every time you see an organelle with double membrane, you know it's pretty important. Mitochondria, its function is it's a fab to produce ATP. We know ATP is the energy source, direct energy source for every living cell. So mitochondria, its main function is to produce ATP. And to turn a glucose, that's a simple sugar, into ATP, there are 3 processes: glycolysis, Krebs cycle, also called the citric acid cycle, and electron transport chain, also called oxidative phosphorylation. There are 3 processes. The last 2 processes happens inside the mitochondria. So about 95% of the ATP in a cell actually come from mitochondria. So that's the main function of mitochondria. And we will go through the whole processes, how to turn the glucose into ATPs in a later structure called the cellular respiration. And its mitochondria is the main place for the ATP to be synthesized. One glucose can produce totally 38 ATPs. And some textbooks say mitochondria used to be a living organism because mitochondria also have their own DNA. That's the evidence say there used to be a living organism. And during the evolution, and mitochondria decide to join the cell. So to increase the chance to survive. And its function is to provide ATP for the cells. Okay, next one, nucleus. Nucleus, we call it the head. When we talk about atom, the atom's head is also called the nucleus. So nucleus, inside you have the DNA. And those DNA, okay, if they are not packed, it's called chromatin. So chromatins are those loose DNA structure. If they are packed together, it's called chromosome. It's called the chromosome. So DNA exists inside the nucleus. So that's the basic nucleus structure. They have the double membrane as well, and they have the nuclear envelope. That's how they call the double membrane. And they have nuclear pore, that's the hole. And when you use the DNA to produce mRNA, that's called the transcription. And those mRNA can go out from the nuclear pore. And once they go out the nuclear pore, outside is the rough ER. It's the rough ER. So that's the place you have ribosome, and they can help translation. So it will go out, mRNA will go out into the rough ER and for the translation to happen. So that's the nucleus. And inside the dense one is called the nucleolus. And those loose DNA is called chromatin. And the envelope, the membrane is called the nucleus envelope. And those hole for mRNA to go out is called the nuclear pore.